right. While people are still showing up, let's just get started. Hi, everyone. Good morning, good afternoon, and good evening for you, and good night for some of you. And welcome to today's webinar, Exploring RNA Dynamics, Innovations in Nanopore-based Native RNA Sequencing, here at our webinar series at Bioprotocol, an open access online journal with a mission to make research more transparent and reproducible. And for those of you who've been waiting for an hour to attend this webinar, thank you for your patience. Just a few comments so you can get the most out of this webinar. If you have questions, you can post them in the Q&A or chat section throughout the talk, and Morgan will address them at the end of it. And this webinar will also be recorded and posted online, so you can also revisit it at a later time point. All right. It is now my distinct pleasure to introduce the moderator of today's webinar, Dr. Ivan Milankovic, who is currently a postdoctoral researcher at the NOVA lab at the Center for Genomic Regulation in Barcelona, Spain, where he also earned his PhD or doctoral degree on ribosome heterogeneity, biological relevance, and translational applications. And by studying ribosomal protein paralogs like RPL3 and RPL3-like, Ivan has identified tissue-specific roles and compensatory mechanisms, and he has also co-developed and applied methods to study RNA modifications using the Oxford Nanopore technology that will be talked about today, including Nano-trap, which couples ribosome pull-down and nanopore sequencing, and Nano-tRNAseq, which is a nanopore-based approach to sequence native tRNA populations to provide quantitative estimates of the tRNA abundance and modifications dynamics. And in addition, Ivan has extensive involvement in science outreach and has served as a head of the biomedicine program at the Petnica Science Center for high school students and completed internships at the Luxembourg Center for Systems Biomedicine and KU Leuven in Belgium, and also won first award in the RIN4 Science Outreach Contest with his presentation on ribosome dynamics, which is really cool to watch. And so with that, Ivan, thank you so much for being here, and the floor is all yours.
You're very welcome. Thanks a lot for the awesome introduction. Now it is my pleasure to introduce Dr. Morgan Lucas, who will be giving today's talk. I'm just going to be here to help out. She's the main star of today's event. So just a brief introduction on Morgan. Morgan's research journey began in Australia, where she is originally from, with a focus on neuroscience before transitioning to cancer research. This early work sparked an interest in sequencing technologies, which led her to pursue a PhD at the Center for Genomic Regulation in Barcelona, where I'm at right now. Under the mentorship of Dr. Eva Maria Novoa, she specialized in developing nanopore sequencing methods to detect RNA modifications in native RNAs and to sequence short RNAs. Now, I have to say, just as a short disclaimer, Morgan and I did our PhDs at the same time. Our benches were one next to the other and our desks in the office were next to the other, so we know each other very well. And Morgan is by far one of the best presenters I've encountered so far. So I don't want to set the expectations too high, but you're in for a treat for today's talk. After completing the PhD, Morgan moved to Munich to co-lead the research and development team at the Medical Genetics Center, a diagnostics laboratory. In this role, she collaborates with colleagues to innovate new diagnostic methods, particularly focusing on sequencing technologies to address a range of disorders. Morgan, the floor is yours, and you have the next hour or so to tell us about Nanopore.
I promise I won't talk that long. Do you see my screen okay? Is it? Yeah, perfect. Okay. Thank you so much for the kind introduction. And yeah, no pressure, Ivan. And also to say all of the work that I present today, Ivan has been a part of. So yeah, this is also his research, and I think he will also answer some questions at the end. But I'm very happy to be here and thankful to Bioprotocol for everything that they do, and also for letting me speak to all of you today, and to Ivan for moderating the session. So today I will talk about basically what I did in my PhD with Eva Novoa in Barcelona. This is the institute where Ivan is currently, where I used to work. It's really this beautiful and this close to the beach. So if anyone is thinking about a PhD, it's not a bad place to do it. This lab, Eva's lab, is an RNA dynamics and epitranscriptomics focus lab, and we're very much interested in technologies and using them to uncover secrets about RNA and its modifications. So a lot of our work is really based on method development, but then also applying this to a biological question. And a lot of what you will see here today is more the method development, because this is what we had to get working first before we could apply it to the questions. And this is really work from the last, I would say, 5 years. I think I started my PhD in 2018. And some of this work I started a little bit before that. So yeah, I'm really happy to talk to you about nanopore sequencing, because I really enjoy it and how it can sequence RNA directly, because I think there's really no other method that can sequence RNA in its true form like Nanopore does. So in case I have to convince anyone why RNA is amazing, I hope that's not the case, because I think it's a really interesting molecule, and the world of RNA is just, yeah, there's so much to understand still. But just in case I do have to convince people, RNA it's an amazing self-sufficient molecule. It can self-replicate, it has catalytic activity, and it's incredibly dynamic. So there are many changes that RNAs undergo during biogenesis, so in the production of the RNA itself, from maturation to degradation. It also changes in response to environmental stimuli. It's a rapid change. In disease, it's also changing, and there's a couple of papers in the last years that show it can mediate transgenerational inheritance of acquired traits, which is really interesting. This is a personal interest of mine. And it's incredibly, the RNAome is very diverse. There's a lot of complexity. What do I mean by this? Well, one, there's a lot of different types of RNAs. We normally talk about mRNA, ribosomal RNA, tRNAs, but there are many, many more types than that. I have been working in the RNA field for quite a few years now, and I still read papers and see new types of RNAs that I've never heard of. So that's really an amazing thing to be working in a field that there's still a lot to be known. And when I say there's a lot of complexity to it, in the biogenesis or in response to stimuli, there are many things that can change. So if we take mRNA for an example, we start with our pre-mRNA, we have a 5' capping step. Then we have splicing, right? So this can lead to alternative spliced transcripts. This gives rise to different kinds of proteins. So there's a lot of variability that can come from just one kind of mRNA. Then we also have 3' processing. So this is poly-A tailing, and the length of poly-A tails can affect many things like RNA structure, how long is it stable for, its localization in the cell. You can also have tails that have uridines, or I think even inosines. There's a lot of variability there. And where I think the most variability comes from, or maybe my favorite level of variability, is chemical modifications. So these are modifications that are decorating RNA. They're on top of the bases, the typical bases of RNA. And I put a note saying it's not necessarily in this order. That's because the modifications can be added co-transcriptionally. They also change in the lifetime of the RNA, like I said, in response to stimuli and disease. So these modifications are always changing. And the epitranscriptome, so this is the equivalent of the epigenome, right? This is the collection of modifications for RNA. It's incredibly diverse. So with DNA modifications, you can see there is still quite a lot. These are the modifications that occur in all kinds of life forms. But when we compare to RNA, there are many more types, right? So there's a really vast range of modifications. And I've marked, this is maybe like a little bit out of date. Maybe there are new methods developed, but overall you can see that there are not that many ways to detect modifications using sequencing-based methods. So we can see red dots for NGS, blue stars for ONT, and triangles for PacBio-based methods. And yeah, really there's a lot that we can do to develop methods to detect RNA modifications. And just a note, the ONT detection methods, when we started using this technology in 2018 or 17, none of these blue stars existed, right? So we developed, and others too have developed methods to detect these. So it's a really early field, and I think there's a lot that can be done in this area. So how do we look normally at RNA sequencing or at RNA? And we talk about RNA sequencing, and this is usually referring to NGS or next generation based approaches with Illumina sequencing. And there are some caveats to this method. So, I mean, it's given us a lot of very important information, but there are some known, let's say, biases that we don't always think about. When we think of biases with NGS, we think of amplification biases, but there are other things going on. How do we do RNA sequencing? We normally start with our template RNA, then we ligate at 3' up to the 3' end. We do cDNA synthesis, so we convert it to cDNA using reverse transcriptases. Then we digest the RNA, so we're left with the cDNA, which is a copy. And then we do some downstream steps like amplification and then sequencing. But this cDNA synthesis step by reverse transcriptases is really dependent on structure and modification. So it's very much affected by these two properties. And RNA is structured and it has modifications. You will see this especially with things like tRNAs when we talk about that. They have really a lot of modifications on each molecule. So these modifications can affect Watson-Crick base pairing, causing the enzyme to fall off the template. So you have short cDNA templates, or it can also cause incorporation of incorrect bases. And then the structure also causes drop-off of the enzyme. So what you see in your cDNA may not be a reflection of what is in the RNA. And when we want to look at RNA modifications, which is an important aspect of RNA biology, we normally have to use another method, and this is often mass spectrometry. And this is a really wonderful method to look at global RNA modification levels. How does it work? We take our pool of RNAs. Here is an example with tRNAs, but it's in any pool of RNAs. And then we digest it up into single nucleotides, and then we look at what is in this pool. And here it's able to really reflect what are the modifications present. So we see that what we see in our pool is shown in the data. This is a very simplified version, but this is quite accurate, and that it's really looking at global modification levels, but you don't know where this modification is in the RNA or on which RNA. It could be on mRNA, it could be on a tRNA, you really don't know, you don't have this context. And the type of RNA and where it is really affects the biology. Then when we look at our RNA-seq data, these modifications, I mentioned, some of them affect Watson-Crick base pairing. So I've just put some stop sign modifications here. And these are the ones that stop reverse transcription. And what that means is that maybe your RNA is not sequenced because it's not reverse transcribed. So there's no cDNA copy, right? And if you look at your Illumina data, you might think, okay, I've only got this tRNA in this example here because it was the only one that could really be reverse transcribed. So if you were to not know what's in your pool of RNA, which you don't, and if you look at just these two orthogonal data sets, you're going to think, okay, this tRNA has these modifications, which is not actually true. So that's why we want to have an approach that can look at both modifications and abundances all in the same workflow. That's why we use Nanopore direct RNA sequencing. One of the questions that was posted was, what does direct mean? It means that we sequence the RNA itself. So we're not doing this cDNA conversion step. We also don't have to do demethylation. Demethylation means we use enzymes to remove modifications. A lot of Illumina protocols do this to make the reverse transcription a little bit easier, but we don't have to do that because we want to keep our RNA modifications to know what's going on there. We can sequence full-length molecules. It's PCR-free, so we don't do any amplification. It means that we don't have amplification biases, but overall it means that we have a simplified library prep, which is nice. You spend less time in the wet lab. And the great thing is that in theory, we can detect all modifications because we're looking at changes in current intensity. I say in theory, we still have a long way to go, but yeah, we understand a little bit better about how we can do this and how the process is. So just a little bit of background of how nanopore sequencing works in case people are not familiar with it. So in this technology, we use nanoscale protein pores. We call them nanopores as biosensors, and they're embedded into this electrically resistant polymer membrane. And then a voltage is applied across this membrane. And this produces an ionic current so that the negatively charged nucleic acids like DNA and RNA, they can go through the nanopore. And then we also have this motor protein that sits on top, and this is controlling the speed of the DNA or RNA. So it's translocating in a stepwise manner, one base at a time. And usually this is around, for DNA, 450 bases a second, and for RNA, I actually forget how fast it is, 120 bases. So there is also differences in the DNA and RNA. And what happens is we can measure the changes in the current intensity in this sensing region. And the sensing region is usually around 5 nucleotides. This also depends on the chemistry. Nanopore has evolved a lot over the years, but yeah, with the current chemistry, I think it's around 5 nucleotides. And what we do is we measure the density of these 5, and then it shifts by 1, then we measure again, and so on. And then we take this current intensity and we base call it. We use base calling algorithms, and this is converting signal into bases. And then what we can do is we can compare modified and unmodified to look at differences in current intensity, or we can use base calling errors to detect these modifications. And we'll talk a little bit about how we do this with base calling errors. But that's the general principle of how Nanopore works and how we can leverage this platform to look at RNA modifications. So today we will talk about EpiNano, which is the first tool that was developed in 2018. I think it was published to detect M6A modifications in native RNAs using base calling errors. In Nanopore direct RNA sequencing, DRS is direct RNA sequencing. Then we applied this approach to look at more modification types, this time in ribosomal RNA, and we could discover new modifications in ribosomal RNA, and then also developed a software to quantify the stoichiometry of modifications, because you can have different levels of modifications in different positions. Then I will talk a little bit about multiplexing tools that are available. And then finally, Nano-tRNAseq, which is a method that we developed to sequence native tRNAs, looking at both abundances and modifications. So let's start with EpiNano, where it all started. So here we used base calling errors to detect RNA modifications. And so it was 2018 that it was published. And this was really a proof of principle method. So we chose M6A because it's a very popular modification. I think this is the most studied of all modifications. And how we looked at this was we first developed synthetic constructs. We call them co-cakes. And they have every single combination of 5-mer. What do I mean by this? So I mentioned before that we sequence, that we measure 5 bases at a time. So we want to know what is the current intensity for every combination of 5 bases that you can have. So it's quite a lot of different combinations. So we made these constructs that have that, so we could sequence them and get that information. So we have an unmodified version, and then we also had a modified version. So we did the in vitro transcription reaction. We would put only unmodified bases, and then in another tube and a separate reaction, we replaced adenosine with M6A. So every single position that has an A was modified. So this was our ground truth. We know we have this modified data set and this unmodified data set, and we can compare them. So we sequenced these, and then from there we took information like the current intensity, the base calling data. So we looked at the quality of the base calls, mismatches, deletions, and insertions. And then we used this data, did machine learning, and then we're able to classify different positions if they were modified or not. So when we look at the IGV tracks here, if you're not familiar with IGV, each bar is corresponding to a base. You can see up here very small, it's AA, GG. And the height of the bar is related to coverage. This is not so important, but the main thing is you see in unmodified, everything is gray. That's because it matches the reference. So it has the sequence we expect. So this is an A and this is an A. In our modified sequence, in our modified constructs, we see a lot of color. And when you have color there, it means that there is a mismatch to the reference. So in here, for example, this should be a C, and we see that maybe 70% of the reads are probably a C here, but then some of them are T. So we can see that the base calling has produced some mismatches, and we can leverage this to look at modifications. So we plotted the base quality, mismatch, deletions, current intensity for all of these, and you can see that there are some differences, for example, in base quality and mismatches between our modified and unmodified data set. And all of these tools I want to mention are on the Novo Lab GitHub, and you can also find all of them on the, I think probably the lab website if you want to use them. So we developed this tool. It was called EpiNano. This is the one that is detecting M6A in this paper. And we, of course, wanted to apply it to something real, to non-synthetic RNA. So we sequenced wild-type yeast and then a knockout strain for M6A in expected positions. So we just did normal sequencing. We looked at the quality, mismatch, and deletions because these are the base calling error signatures that were important for M6A. And we could see that there were differences. And if we look at our IGV tracks over here in the top, for example, for the 3 replicates of wild type, we see at this position, this is the expected M6A position, that we see green here. So there is mismatches occurring here because we have the modification, and in the knockout, it's gray. So that's because there are no base calling errors because there's no M6A there. So we could successfully detect M6A in this way, which was really exciting for us. And then it led us to a lot of new things in the lab. So one of them was looking at ribosomal RNA modifications. We did the same approach. We took our cocoa cake molecules, constructs, and then we just in vitro transcribed with other modifications. We did pseudouridine, M5C, HM5C. We just picked ones that we thought would be cool, might be interesting. If we could in vitro transcribe them, then we wanted to sequence them and see how it looked. And something that we noticed in our early work was that the modification, the reads with modifications had differences in mapping and base calling. So here we also benchmarked the effect of which base caller or mapping algorithm you use and which parameters you use. There was another question about what's the difference between Illumina RNA-seq data analysis and nanopore sequencing data analysis. You still do a lot of the same steps, mapping quantifying modifications or whatever downstream things you do. But it's really about what kind of algorithm you use and what are the parameters, because the data is so different. So we had to play around with these, work out what really worked for our data, because you can see with, for example, reads that had pseudouridine, if you use particular mapping settings, you would lose all of your data. So we managed to play around with this so that we can look at our in vivo data sets. We also looked at how this affected the base calling qualities. So these are all the same sequencing data, just analyzed with a different base caller, and you can see there are differences, right? So it's really the choice of base caller and mapper is really affecting your analysis. So this is just to say, when you're working with new platforms like this, it's really important to think about these things and try and benchmark them to make sure that you're not losing data. We were systematically losing modified reads. And if we wanted to look at modification levels in, say, disease versus not disease, we would have probably not seen anything because we weren't capturing them. So we did a lot of work to get this working. And when we were finally happy with what we had, the choices of algorithms and parameters, we sequenced yeast ribosomal RNA. And this is just some IGV snapshots of the different ribosomal RNAs, and you can see the modifications where some of them occur. You see these strong base calling errors. So we thought this is a pretty good sign. We'll be able to detect some of these with base calling errors. So we wanted to look at the signature, right? So what kind of mismatch frequency, deletion frequency, quality to each modification give? Can we distinguish modifications from each other in the same RNA? And we could. So we looked at the typical modifications that are in ribosomal RNA, which there is a lot. It's a very modified molecule. We have pseudouridine, M1A, M5C, AC4C, and these NM modifications. And we saw, for example, for pseudouridine, it had a really, really strong mismatch signature. So we saw that it had what we classify as a U to C mismatch. So if you are just sequencing your sample, you would normally expect to see a U that's base called, but it would be giving a C instead. So this was a sign that actually there's a pseudouridine in this position. So we wanted to, of course, validate this. And one way that we validate these results is either to do this in vitro transcribe with modifications or not, or to use knockout lines again. So here we have yeast lines that have knockouts for snow RNAs. Snow RNAs are guiding the modification position. So if you knock out particular snow RNAs, then this modification doesn't occur. And we did this for pseudouridine and for these NM modifications. So we saw, for example, in this knockout, in the expected position, that we lost this base calling error signature. So this is what it should look like. And here it's gone. And then the same for all of the knockouts, it was in the expected position. And these blurry plots are showing the same thing, that we see a decrease in the mismatch frequency in our knockouts because we lose the modification in the expected sites. So this was a really good sign. Then we wanted to take it a little bit further. So Leszek, who's a really amazing bioinformatician in the lab, he developed Nano-tRNAseq, which is a software to look at the stoichiometry. So really to say how modified is this position in my RNA. So he spent a lot of time on this, and I cannot explain all of the things that he did in this seminar. You can look in the paper, but it's a really great tool. And I think there's a second version coming out at some point, or maybe it's already out, but you can see at Novoa Lab GitHub all the tools that are there. A lot of them are coming from Leszek. And then from this was all in the same paper. We were able to use these methods to predict or to identify new modifications that had not been identified before in ribosomal RNAs in yeast. So for example, we found here in position 854 in the 15S mitochondrial ribosomal RNA that it seemed there was a pseudouridine there. That's not documented. And when we look at the structure or the position where it is, it seems to be in this loop here, and it has this particular motif. And this looks very, very similar to this tRNA T-arm that has pseudouridine position, I think it's 55. And we know that this modification is added by this enzyme PAS4. And you will see this the same diagram a little bit later, because we use the same knockout lines over and over because we have them in the lab, so we use them to validate different things. But we thought, oh, maybe this is the same enzyme, the same mechanism. So we sequenced the knockout and the wild type, and we saw that we lost the signature. So in this case, we could identify unknown modifications in yeast ribosomal RNAs, and we could actually validate the mechanism that they're added. So we could say that PAS4 is the enzyme that's adding this modification here, as well as in tRNA that's already documented, which was really a nice biological finding. That was a positive result. We also had plenty of negative results in this paper, and I think it's really important to show those things too. Negative results are as important as positive results. And we did a lot of things with oxidative stress, thermal stress, just doing everything we can to see if there are differences. We didn't see any differences there. And also Ivan did a lot of really nice work on getting the different fractions in translation, and we looked at the modification changes there, and we did not see any changes in RNA modifications in ribosomal RNA across the translational repertoires. But it was nice to be able to look at those things. So that was our modification tools, and by this time we were sequencing a lot of different things, right? We were trying to look at different RNA modifications in different species of things, in different settings, in disease or oxidative stress, whatever. So we were sequencing a lot, and there is currently no method from Nanopore for multiplexing RNA runs. They have it for DNA, but they've never released it for RNA. Right now is the Nanopore Community Day in Boston. So they're going to be releasing new things in, I don't know, maybe 4 or 5 hours. Maybe there's going to be a new tool for RNA multiplexing, but they've also been saying that for years. And yeah, this doesn't exist. So we had to develop our own because it's very expensive if you're always sequencing one sample at a time. So this work was published in 2020, and I'm really glad that it was, because it meant that we could sequence many, many more things in the same flow cells, and it works very well. So this was a tool called Diplexicon. Again, it's at the Novolab GitHub link. And I did the wet lab. I really didn't contribute anything to the bioinformatics. Here it's the amazing work of Martin Tansel and James, and also Juan Lee. So what we did here is we developed these barcodes. So barcode 1 is exactly what you get in the kit when you purchase a Nanopore direct RNA sequencing kit. And it's a DNA barcode. So when you sequence DNA in an RNA library, the signal looks very different, right? So you can see this very big drop, and this is the current intensity over time. And this is as the molecules going through the pore. And you see there's a big difference. So you can segment that section of the barcode. And we just took this barcode and rearranged it. So we had four different barcodes, and then we sequenced different things with these barcodes in different flow cells, and then gave the data to these guys. And what they did was they developed a way to segment this particular part of the signal and then convert that current intensity into an image, so into an array of pixels. And then they use this for deep learning to be able to classify which barcode. It's really very, very cool work. And yeah, like I said, it was until recently the only tool available for multiplexing. There is a paper on bioRxiv called Warp DMAX, which is another RNA multiplexing tool. It's by Veep and Patrick. I also encourage you to look at that. And there's also another tool coming out from Novolab. I cannot give any details on it because I also didn't work on this, but I just wanted to highlight that there's more coming in this area. So this is the work of Gregor, who's a PhD student in the lab, and then of course Leszek, and then with Eva and other people in the group, but they've developed a way to multiplex 100 samples, which is amazing. So going from 4 to 100 is huge. And more importantly, so this is comparing, it's called SeqTagger, this approach. This is comparing it to Diplexicon with different confidence cutoffs, but we can see it has a higher precision, a better recall, but more importantly, it's just much, much faster. And I think that because it's so much faster, maybe at some point they can do something like barcode balancing. Let's see if they do it, but I would really like them to develop this. So basically, while you're sequencing, if you realize one of your samples doesn't have as many reads as the other, you can switch on this tool and it's able to selectively sequence more of that sample than the other. And this is in the flow cell, not in the library. So it's something that you can do live and interactively. This is a really nice thing about Nanopore, is this adaptive sampling approach, which I won't go into here, but I think this is a really, really cool and useful area. So finally, I will talk a little bit about Nano-tRNAseq, which is a method for sequencing tRNAs, looking at the modifications and abundances. And yeah, this project, it was published last year, but it took really 4 years to get it working, and it was really not working for a long time. So I hope that when anyone sees this kind of work, they're not thinking, oh, you know, I don't know, I have to, how do I say, it took a long time, a lot of things went wrong, and just because there is this paper doesn't mean that there wasn't a lot of failure along the way. So it's, I just want to say this because I think it's really important when you look at these kinds of presentations. I'm showing you all the good things and all of the publications, but there are many things that don't go right. So yeah, but let's talk a little bit about tRNAs. So as we know, they're very important molecules in decoding genetic information, but they're also very involved in disease, right? So if you have changes in their abundances, modifications, and the way that they fragment, actually this affects many, many diseases. I don't know if this is a very known area, but I find it really interesting. And I think there'll be a lot of future in tRNA therapeutics. And tRNAs, they are really a crazy molecule. They're so modified and they're so structured. And we thought naively, if we can sequence these, we can probably sequence any RNA type. That's not necessarily true, but we tackled this because we were interested in tRNAs. Eva has a really long history in tRNAs, and so we thought, why not? So what do I mean by structured and modified? This is a secondary structure of a tRNA. In green, I've put every site that might have a modification, a possible modification. Of course, it's changing a lot during biogenesis and during the evolution of the tRNA or the state that it's in. But these are the positions it can be. And then these are the positions where we have these modifications that affect reverse transcription. And you can see it's a lot. And on average, one molecule has around 13 modifications. And I really like this quote from Francis Crick about how tRNA looks like nature's attempt to make RNA do the job of a protein. It's really an unusual RNA molecule because it also has this very compact tertiary structure. And this, of course, really affects sequencing. It's very hard to ligate things to this structure or reverse transcribe it because it's really, really compact. So yeah, there's a really complex interplay between tRNA abundance, modifications, structure, which really dictates the function and the biological role of these molecules, but it makes them very difficult to sequence. And we talked about this before about how we do RNA sequencing, and I mentioned that structure and modifications really affect this process. And for tRNAs, it's really impossible to use a normal RNA sequencing method. You need to do something. There are a lot of protocols out there, ones that use demethylases, so they remove some of these modifications that cause problems. There are ones that use some kind of hydrolysis to break up the structure. And there are other ones that improve the reverse transcription and also the bioinformatics tools. And these are really good, but they all have their caveats, right? So you can get a lot of different biases. You lose information when you're changing the tRNA so much. So we did it with Nanopore, and we had to do a lot of different changes. It's in the paper, a lot of the things that didn't work. Again, I think it's really important to show the things that don't work, because I don't want people to make the same mistakes, right, if you can just read it. So I really encourage people to try and put negative results in their papers if they can, if they're allowed. I'm very lucky Eva is all for that. So there is a lot of section in the supplementary that's dedicated to the things that didn't work and why. But at the end of the day, what we found is that we had to tackle the length of the molecule, the structure, and the modification. So pretty much everything. And we tested all different combinations we could, but there were a few things that we found that really worked. So one of them that's quite important if you want to get the full molecule is to extend the ends with RNA. If you do it with DNA, it doesn't really work because it cannot be base called. You saw in this demultiplexing plot that the signal looks very, very different. It drops much further down than RNA. It's a very different signal. We cannot really work out what's the sequences there. So we need to ligate RNA. And why this is important to extend the end is because when the molecule is being sequenced at the start and the end, so you can see it here, it doesn't have the motor protein. So at some point, the motor protein flies off because it's not attached to anything anymore. It's not attached to the nanopore. It's just sitting on top because of this current, and this RNA is pushing it on the molecule, on the nanopore. But when this RNA is most of the way through, it flies away, and then you're sequencing the last 15 bases without any speed control. So you pretty much lose that information because it's not normally translocating, so the base caller cannot really get that signal. So we tend to lose about 15 base pairs on either side. That's why we need to put RNA, so that we are instead losing the adapters instead of the tRNA itself. We also looked at what lengths we need to do, because RNA oligos can be expensive, right? So we wanted to have it as cheap as possible. So we found around 25 bases is really like, then everything, you get the whole molecule and it's not too expensive. We also tested a bunch of different reverse transcriptases. So we don't sequence the cDNA with direct RNA sequencing, but we can use it to linearize the molecule. This just helps with the translocation speed a bit. And again, I tried many, many different ones. There was a paper that came out a year after this paper, I think, where they did a much better reverse transcription than I did. I recommend to use theirs if you're going to do it, but we also found things that work for us here. And we found that it increases our sequencing yield by 1.5, and we don't get any biases. So when I compare the abundances of tRNAs with and without reverse transcription, it looks very normal. So we're not biasing anything. By doing this, we just increase the yield. If you don't want to reverse transcript, do the cDNA synthesis, it's not necessary. And the big thing that we found out was that our sequencing software, Minnow, this comes from ONT, it was filtering out all of our tRNAs. So when you do a normal sequencing run, it looks like this. It's all green. Green is good. It means it's sequencing an RNA. We have 1 to 2 million reads. I think it's much more now with the new chemistry. And I was getting around 20,000 reads. And this is an overestimation. I had some runs where I had 4,000 reads, and it was really sad. I think Ivan probably remembers a lot of lab meetings where I would just go and complain about my very sad sequencing runs. And what we noticed was that we had a lot of yellow pores. So each square here, this is just a picture on my phone of the sequencing software. Each square here is a channel on the flow cell, and it's saying, oh, you're sequencing a lot of adapters. I'm like, well, I didn't put that many adapters. Probably it's tRNAs. How it works with the sequencing software, it's constantly monitoring the signal intensity. over time, and then it decides when it's a genuine read based on some criteria. And that's very smart normally, because you don't want to keep all of the information, right? You don't want to have millions and millions of reads that are just adapters. You want to filter them out. Otherwise, your analysis and your storage will be just too long and too high. So that's normally okay unless you have short RNAs. So we managed to find a way to work out what do we need to change in that configuration to allow us to keep the reads. Because if you sequence normally with Minnow, it filters these reads and you never get them back. So you can never go back and find those reads. It's outputting only what it classifies as a read to the FAST5 file. So this was with Leszek, who figured out we need to change the adapter time and the strand time. This information is not so important. It's just that we could classify, we could retain the tRNA reads. This, of course, improved our sequencing yield by quite a lot. But the most important thing is that we could recapitulate known tRNA abundances. So when you sequence with the default settings that Minnow gives you, you have an overrepresentation of long tRNAs because they're easier to sequence because they're longer, and then you have an underrepresented representation of short tRNAs. So this one is 68 versus 93 here. And this is a plot just showing that we see observed versus expected read counts when we use our custom setting. And here this is showing the same thing, that it's much closer to what we know, what we expect. Then next we wanted to look at modifications, if we can see that. So this is the PAS4 line again that we had from the original paper on our ribosomal RNA modifications. So we said, okay, let's sequence that. And we can see at position 55 in the wild type, we see our pseudouridine signature, which is pretty strong here. This is a zoomed in version, and it's missing in our knockout line. This was what I was talking about with the paper, the previous one. And of course, we confirmed looking at the base calling errors. If we look at the wild type that has the modification, that it's a little bit, or it's much higher than in the knockout line. And we see this in the expected position. So it's not just changes in global pseudouridine, but in the position 55. And the really, really cool thing about this is that we can look at the whole tRNA, so along the whole, every position of the tRNA, and also in different ISO acceptors, right? So we really know what is changing and where. And this is a plot of signal base calling, summed base calling errors relative to wild type. So this is the PAS4 knockout line compared to wild type. When it's red, it means there is an increase in a modification or something, and blue is a decrease. It's a decrease in the base calling error. So in PAS4, we expect at position 55 that we have a decrease in base calling errors because there's no pseudouridine there. So we want it to be blue, and we see that. But we also see there's a lot of blue going on around it. And we were like, okay, something else is going on. There's other changes. And for a while, we thought we discovered a modification loop, and then we realized it was actually published some time before. So this loop is known. We didn't discover it, but we could just show it in all tRNAs. So in this paper, they were able to show it in one ISO acceptor, and I think it probably took quite a lot of time. Whereas here we can show it in all ISO acceptors, and we can look at global changes and modifications. So what happens is pseudouridine, PAS4 adds pseudouridine 55, and this allows the addition of these other two modifications. And when this is lost, they cannot be added. So we see in particular tRNAs, not in every tRNA, that this is also changing. And we confirmed that it was in the expected tRNAs. And then lastly, we also could look at deadenylation. So we put the yeast in oxidative stress. I think, Ivan, you did this. You were doing all the yeast experiments. So what happens is you see this very striking red line here at the end of the tRNA, the last position. This means an increase in something. And what is happening here is that we have a loss of this terminal A. So we see a deletion, and that's because it's being lost. And this is also something that's been observed in one ICE acceptor, but we could show it in different ones here. We could also look at the deletion frequency. And we think this is a very rapid way of changing amino acylation. If you don't have this A at the end of your tRNA, then the amino acid is not added, so you can repress translation in a stress state. So we think that's pretty neat that we could also see that here. So this paper was published last year, but there's a team of people that work on Nano-tRNAseq now since I left Novoa Lab, and they're doing really cool things. This is just a summary of what I showed you, but it's also working in, so I did everything in yeast, but it works in mice, bacteria, human samples. There's multiplexing as well. The tool was patented and licensed to Imagina, so they're also improving. It's a service that will be available as a kit, which should be a much improved method. There are some caveats to the method, of course. For now, it's just looking at the difference in modification. So you want to look at healthy or disease or knockout or not. But in the future, when we improve our analysis, that won't be necessary. The same with needing prior knowledge of RNA modifications. When we understand how these signals look in tRNAs, we won't need to do this anymore. We'll be able to do it without prior knowledge, so we can discover new things. And there are many applications for this, probably more than I can ever think of. But first of all, looking at the tRNA modification profiles in yeast, it's really described, but I was really surprised to learn, because I didn't know much about tRNA before I started this. In humans, we only know the modification profiles of 18 out of 200 of the tRNAs. So probably there's a lot to learn there. Also looking at different modification loops, because they're all interconnected. Looking at tRNA acylation and charge. There is a paper from Laura White, who actually developed a method so took Nano-tRNAseq and then added some steps to look at acylation and charge of tRNAs, which is really very, very cool. And the lab are also looking, they're testing if this can be used for a diagnostic tool, which I think is really relevant because it's involved in so many diseases. So yeah, I wanted to thank everyone, because of course this is not just my work, but the work of many, many people, the whole lab, including Ivan. But yeah, it was really a great time to be working in this lab and working on these projects. It was very frustrating sometimes, but very fun. And it was nice to work in such a supportive environment. So yeah, if there's any questions, I would be happy to take them. Sorry for talking so long. I think I just missed talking about nanopore and RNA. So yeah, you have to tell me to stop talking sometimes.
Thanks a lot, Morgan. As always, an amazing presentation. There's quite a few questions around here. So if you don't mind, I can start with them from the Q&A section. I'll start just chronologically, and then we can go through them one by one. So the first one was, are there any studies correlating the presence of modified RNA and altered splicing?
Yes, there is a lot, particularly with M6A. There are lots of studies related to M6A and alternative splicing, but I imagine that other modifications that are in mRNA are also involved. And this is a way to be able to look at that. Nanopore sequencing is a great way to look at alternative splicing, but being able to do that with modifications on top, I think there's a lot to be understood there. Yeah, good question.
The second one is about, I think you will like this one. It's about single cells. It says, what is the amount of RNA required for each experiment, and can it be applied to single cells?
The amount of RNA that's required for experiments, it depends a lot on what you want to sequence. I know I hate when I ask a question and someone says that, but it really does depend, because if you're sequencing shorter things, you probably need less, and longer things more. Yeah, it also depends if you're looking for certain things within a pool of RNA. I would say for tRNAs, they got down to 100 nanograms, I think, for library prep. but a lot of nanopore library prep requires 500 nanograms. So it's quite a lot compared to Illumina-based protocols. For single cell, this is definitely something that's in the works. I am not working on single cell myself, but there are people in the lab and in other labs that work on it. But I cannot tell you how much they need for that. And I think this is a problem with direct RNA sequencing. I think it will be very, very challenging. I think a lot of people go the route of using cDNA sequencing with amplification for Nanopore. So this is similar to Illumina in that you do the conversion, so you do lose information about modifications, but you do have the advantage in that you have the full-length molecules. So long-read sequencing is beneficial to short-read sequencing in that you can read to very repetitive regions or look at large structural variants and things like that. So it's still losing information, but yeah, I think there is potential, but I think it's going to be really difficult for single-cell RNA-seq, direct RNA-seq. But I hope that I'm wrong.
Okay, then there is this next one that I think is very relevant for some of our early efforts in the lab when we were setting up these methods. The question says, do all base calling errors correspond to modifications? And if not, then what is the error rate?
Good question. No, not all base calling errors correspond to modifications. It could be that you have maybe something that's very structured that affects the translocation speed. There are many, many factors, and this has evolved a lot over time. So this technology was released in 2017, and it has gone through many iterations of chemistry and base callers. And some, when they release a new base calling model, sometimes it doesn't detect the modifications anymore. So yeah, it's kind of tricky. Like we were doing it within the context of a particular base caller, and we would know how that would behave, but this really changes with the base calling chemistry. So what I really think, this was a very good approach at the time. It's what we had, but I think in the long run, what we really need to use is the current intensity only. But this was very difficult in the early days when it was much less stable. So for reference, the error rate of Nanopore direct RNA sequencing used to be like 15 or 20%. Like it was really high, really, really high. Now it's 96%, depends on the base caller as well, but I think it can be up to maybe 99, but it's changed a lot, right? So yeah, not every base calling error corresponds with a modification. Not every modification gives a base calling error. But my idea or my thought on this is that we really need to look at the signal intensity going forward when the technology improves. Yeah, really nice question.
Do you also think that then it makes sense to always have a control that you're sure is completely devoid of modifications, such as an in vitro transcript, for example, for ribosome RNA or for a protein coding gene that you're interested in, because then you're gonna know that if you have errors there, they're not gonna be coming from modifications, but more likely from sequence contexts.
Yeah, yes, definitely. When you're developing the base calling algorithm that can detect modifications from signal intensity, you need to have this ground truth data set that's really like, this is definitely modified and this is definitely not. This is really essential. But then this is true not just for Nanopore, for any technology where you're developing some kind of base calling algorithm. And this is sometimes the challenge, is to get this ground truth data, particularly for tRNAs. But yeah, once you have it in that context, in that chemistry or software, it should in theory be able to be applied to other settings without having the control data set. But of course, when you change to a new sequencing chemistry, you need to revalidate that again with modified and not modified. Yeah.
Cool. Thanks. Just one thing I would like to ask the participants to, if possible, ask questions in the Q&A section rather than the chat, because it's a bit easier to go through them. But if not, I will go through the chat as well, so no worries. The next question has to do with us as ONT customers. So it says revisions of direct RNA sequencing kits are made sometimes, and previous versions tend not to be available. To what extent, especially for machine learning based tools, does this cause problems with modification calling? Is it a concern for new projects?
I don't know, Ivan, do you want to take this one, or because you're
I mean,
also working this?
I can, yeah. So the thing is that what happened very recently was that basically the chemistry that was used for direct RNA sequencing was discontinued, and you can't buy it anymore. So the RNA 002 flow cells are not commercially available anymore. So most of the work that Morgan presented here was done on these flow cells. And then some of the tools that were, especially the machine learning based tools that were developed on this chemistry are basically not, they don't work for the new chemistry anymore. So they're going to have to be remade, redone, right? So this is a bit of an issue, and the bioinformaticians who work on this are going to have to redo the effort that they have already done in the past. And it kind of was unexpected that ONT would do this, that one whole chemistry would be discontinued, and then that we would have to retrain models for the new chemistry. However, I have to say that this new chemistry is a lot better than the previous one in terms of coverage, in terms of the input amounts, etc. So hopefully that now when we already have the knowledge from the previous chemistry, we can use it for this one, we can train models, and we can move on from there. So I don't think this is a huge issue, but it is a bit of a complication.
On this note, maybe I can offer a perspective from diagnostics. So now I'm working in a diagnostic laboratory in Munich. And these changes in chemistry, it's very, very challenging. So we cannot just retrain something, and we have to really validate that it works the same or better. And it has to be done to a specific standard. And this takes time, and it takes money and people, because for every, I don't know, variant that you're testing or every gene, you have to make sure that this new kit is behaving in the same way. And often you also have to adapt the protocol. So I worked a lot recently on the change in chemistry for the Cas9 kit for DNA. And yeah, in a commercial setting, if you're using these for diagnostics or prognostics or something, it's really challenging. So now Nanopore have released this Q line where they keep everything stable for a longer time. So as Ivan mentioned, they discontinued chemistries, and this can be within a year, right? And projects, not just in diagnostics, but in research, my PhD was 5 years, right? So if they changed chemistry during my PhD, it would have been really difficult. I was lucky that I didn't have that, and now everyone else has to do that. Ivan has to change everything for the new chemistry, but yeah, it can be difficult. So there is the Q line available, which is maintaining the same products for longer. But then I think for RNA sequencing, we want to have these better chemistries, right? Because it's still really early stages. So we want to have better coverage and more accurate results. So yeah, it's annoying, but it's also good that they improved their chemistry, yeah, and algorithms.
The next question has to do with RNA biotypes. So just as a bit of a backstory, in our lab, we were kind of interested in all different kinds of RNAs, but this was split between different people in the lab. And Morgan and I were mostly working on tRNAs and ribosome RNAs. But this question from Steve has to do with mRNAs. So Steve is interested in whether, since mRNA is less abundant than tRNA and ribosome RNA, whether you have any recommendations for specific enrichment beyond oligo-DT-based methods.
Unfortunately, I think oligo-DT is really the best. Something that I did when I was working with longer RNAs, because tRNA is a really big fraction. It also depends on what you work on, but for example, in yeast, it's huge. Like there's so many tRNAs. I just would do a column cleanup with changing the ethanol percent so that I removed maybe things less than 200. This definitely helped a little bit with enrichment. But I think for mRNA, oligo-DT is really, Yeah, it's very good. You can, of course, just take your input RNA and do a regular library prep, and because it already has the poly-A on mRNA, it should be the thing that's ligated and sequenced in Nanopore. So you can do enrichment like that. I think there were comparisons between doing oligo-DT enrichment versus this method of just doing the library prep on the total total RNA, but I don't remember if it was much better or not. That would probably be nice information for you, Steve, but I don't remember, sorry. Yeah, we did a long time ago try digesting ribosomal RNA using hybridization of DNA oligos and then using RNase-H, I think it was. Awful idea. It really fragmented everything. This is okay if you do Illumina with Nanopore or PacBio. The integrity of your RNA is super important. So you need to be really careful with your handling. And we always found that oligo-DT is really the best. But you could also give a try to just doing a column cleanup or whatever kind of cleanup and adjusting it so you lose the shorter RNAs, like less than 200, gets rid of your tRNAs, and then trying just to prep on without the poly-A step of the library prep and just using the native poly-A of mRNA. Yeah.
Thanks. The next question says, are there any ways to chemically modify certain bases similar to bisulfite sequencing for enhancing the detection by Nanopore?
Yeah, definitely. You can use enzymes for this, and you can also use chemical probes. There are lots of different ones out there, and people use this for structure studies in RNA. Yeah, so you can convert different bases to different modifications using these things. The thing is that you're never sure what the conversion rate is. So it's never probably 100%. And when you do your when you're trying to develop these base callers or algorithms to quantify modified and non-modified, you want to be completely sure that you have a modified position and an unmodified position. So this is why we tend to not use these approaches to do that. But it's definitely an approach that is used for structural studies because these probes often only change the base when it's unpaired. So you can look at unpaired and paired RNAs. Yeah.
Thanks. For the sake of time, I will skip one question by Steve so that we have other people asking questions, and I'll go back to it if we have time later. So there's a question by Kandar who says, hi, Morgan, excellent presentation. One naive question. Can you distinguish between tRNA and TRF transcripts in your Nanopore data?
Yeah, this is a really nice question. You can, if you have a full-length molecule, of course, it looks like a tRNA, but we can also look at if the 5' and 3' adapter is ligated, so we understand really if it's intact or not. And with the tRNA fragments, we are able to capture them, and we know that we're able to capture fragments, and we've also done tests on like in vitro transcribed tRNA fragments. But with the Nano-tRNAseq method, it only captures 3' tRNA fragments because it's relying on the CCA overhang. So for looking at fragments, we use a different approach. This is something that's in the works, but yeah, we can definitely look at it by looking at this 5' adapter that's ligated and mapping that and saying, okay, this is a full-length one versus not a full-length tRNA. Yeah.
Thank you. Then Rodrigo was wondering if there is a bias in the tRNA sequencing against the highly modified tRNAs, especially related to the mapping, as those would have more errors. And then related to that, is there a way to validate the calculated relative tRNA abundance?
Yes, we definitely in the early revision, like with the mapping especially, saw differences with base calling. It was okay. The later models of base calling is really very good, but we can base call all reads, but mapping we would see differences. And this was probably the most challenging part of the entire project, was to get the mapping working. I worked on that for years to get something kind of working, and then Leszek, who is 100 times faster than me at bioinformatics, came in and further had to improve it. And also we published something that worked, and then we also found better ways of doing it. So the mapping is really challenging. Yeah, so maybe there are some biases, but we also see that we capture very modified tRNAs. So we know at least for yeast, which positions could be modified. And we know that we are capturing tRNAs that do have all of those modifications, but we cannot be 100% clear about the bias, right? Unless we actually make a sequencing pool that has known tRNAs with modifications. But about the relative abundance, we did we are able to validate that because we took in vitro transcribed tRNAs that don't have modifications, and we made sequencing pools of very specific amounts. So we did equimolar amounts, non-equimolar amounts, many different combinations to make sure that what we sequenced was what was in the tRNA pool. So we can do it for tRNA abundance. It is a little bit more tricky when you have modified tRNAs because their modifications are changing dynamically, and there's a lot of different variables there. But for abundance, we definitely did validate this in the paper. And this was a big part of the paper because there is another method that sequences tRNAs, and they never validated the abundance. So they never realized that they were losing all of their short RNAs, right? So yeah, with that, we're pretty confident, but it's true that the modifications can cause problems. Very nice question.
Thanks. There is another one about tRNA modification interdependencies and how they measure, and whether the error rate is consistent on specific modifications, and whether it provides a specific signature.
How does tRNA modification independence? Okay, how does it measure? So is that in terms of like, how do we measure this? So we can see in, for example, we take these knockout lines. This is how we were identifying them, as taking these knockout lines that knock out one modification and then see how it affects other positions in the tRNA. So we are able to see all the positions of each isoacceptor of the tRNA. Is the error rate consistent on specific modifications, and does it provide? Yes. So this is something, particular modifications do give specific signatures, error signatures, also intensity signatures. And this is the challenging part, is to actually find out what that is. And in tRNA, it's especially challenging because you're trying to do that when you have other things in the same area that can be very, very close, that's also changing. So yeah, we do look at specific signatures, but we also in tRNAs have to look at the signature in relation to other other modifications, which is very difficult, but there are people working on that in the lab. So I hope that they manage to really find all of these signatures in tRNAs, because there's so many modifications that are occurring and changing.
Yep. Thanks. Then the next question is from Victor, who's interested in mapping protein binding sites globally. They use UV cross-linking to fix protein RNA complexes, and after proteinase K treatment, peptides remain attached to the RNA. Could it be appropriated to interpret any errors in base codings as peptide attached sites? Could these peptides block the pores?
I once we did the calculation for this, right, Ivan? About if it blocks the pores. I think it would. Yeah, I think it. Yeah, it would block the pore. Unfortunately, yeah. I think for amino acids it's okay, but for peptides, unfortunately, it would. I don't know if you did something like this, right, where you do cross-linking and then you digest the regions around it, like these footprints, like ribosomal footprinting. Wouldn't this be something kind of like you could do the cross-linking, digest the RNA around, and then remove the attached protein and then look what's there? I don't know if this is
Yeah,
something
but
to do.
there were no
Hmm.
covalent bonds between the RNA and peptides. I think this would
Yeah,
be a bit of
maybe it's very,
a
very
deal breaker for
difficult.
nanopore. But then
Yeah,
it's hard to tell. I mean, it really depends on the size of the peptide, so one could try, but
yeah.
I think that in our hands we had pores clogging with less. I mean, there are
With
yeah, even
tRNAs.
even even tRNAs which are highly structured,
Yeah,
they can clog the pores.
yeah,
So
yeah. Sorry, Victor. Maybe it's not good news.
then there is one question about flongals. Is it possible to adjust the direct RNA sequencing protocol to make it working on flongal when you only want to sequence short non-coding RNAs instead of the whole transcriptome?
Yeah, definitely. Some of some of the work in this paper was done on flongos, actually. Yeah, because the sequencing runs were so bad, I could not justify or convince people to use whole benign flow cells for this, so I used flongos. And this is definitely feasible. Yeah, and flongos are much better than they used to be. So I suggested if you want to do a smaller subset or you want to do a test to confirm something, try flongos.
Thanks. Then there is one about how fragile RNA modifications are during the library preparation. So Vadim is wondering, what are the objects you used for the results presented? And he says, my guess is that the modifications are not so stable and may be lost during isolation and etc. So all the procedures are prone to errors or need special care.
I guess this could be a possibility, but for example, in our IBT RNAs, we saw because we know where the modifications should be, and they were always modified in those
Yeah,
positions. So in that
I would say
case,
this.
yeah.
Mm.
And Ivan, maybe you probably know better as you're more of a chemist than I am about the stability of RNA modifications during the preparation.
I would say it really depends on the type of modification. I was mostly working with
Mm.
ribosomal RNA, and there's like 200 something annotated ribosomal RNA modifications. And most of them were easily detectable in most of the samples I've ever worked with, and they undergo harsh treatments during library prep and during isolation where you use trisol, etc. So I would say that most of the modifications that we know of are stable enough to be identified using Nanopore. Now, if there are
Yeah.
other modifications that get lost in the meantime, both during Nanopore library prep and Illumina library prep, then this is hard to answer, right?
Yeah.
Even mass spec is also even probably harsher.
Yeah. Yeah, I tested a lot of different extraction methods, library prep methods as well, like doing overnight ligations or different ways of extracting and these kinds of things. And the I mean the abundance, like not the abundance, but the amount of RNA I got would change, but never the modification profiles or abundances. So at least in those conditions that we tested, yeah, but as Ivan says, probably there are things that changed, but it's true in all library preps, unfortunately. So we can never really be totally sure about that. Yeah. But at least in our in vitro transcribed ones, we know what there is and it never changed. So hopefully that gives me some hope that it's not changing too much during the handling process.
Mhm. There is one question regarding deep lexicon. So when you add the DNA adapters to the RNA and the reverse transcription step comes next, does it mean that you're sequencing the DNA as in the Illumina protocol?
Yeah, so the DNA adapters also serve as the reverse transcription adapters. So this is where the reverse transcriptase is kind of, I don't know, binding and then doing its thing to make the cDNA. But then we are sequencing it, yes. So we see it in the signal and we sequence it. This is part of why we could extract it so well, because when it's sequenced, when surrounded by RNA, it looks very, very different. So yeah, we are sequencing it, but it's kind of like it's attached to the RNA and then it goes straight to the RNA sequence. So it's all sequenced in one. Yeah.
Then I'm going to go back to Steve's question that I skipped. Oxidative stress has also been noted to cause tRNA cleavage in the anticodon loop. Did your group consider or observe this phenomenon? Some of the fragments themselves have been observed to have an impact on translation.
Yeah, we learned about this, I think after this paper was published, or I mean, even quite recently. And I think we've never really looked at this, but there were some, I remember some protocols that were even suggesting to do like a some kind of like nicking step, like that you can ligate those nicks in the tRNA from different stresses and then sequence it. But I don't think we've actually done anything concrete on this, but I think it's really, really interesting. That's for sure. Yeah, I don't know, Ivan, maybe you have more of an update on this.
Not really.
No?
I know as much as you do. Okay, there's a few other questions from the chat box. Basically, these tools work based on base cloning mismatch. How to apply these tools for complex plant genomes, which are polyploids and heterozygous?
How to apply them for complex genomes? Okay.
Yeah, so I guess maybe this would be a bit of a different question because
Yeah.
you were not really focusing on
Yeah.
DNA during your
Yeah.
PhD,
So the good news is I work on DNA now and things like methylation, and it's so much more advanced than the RNA tools. Because I think, we were RNA kind of came after in interest for sequencing. Once you get the DNA sequencing working, then they move to RNA. And so with DNA sequencing, they don't use base calling errors at all. They really have very good modification where base callers that are very, very accurate with nanopore data for genomic data. So I think you don't have any of these problems. You can do your sequencing, and then from the base call data, you can use I think Megalodon is probably the best tool to use. This is what we use at least. And then you can call directly, and all this training was based on the signal intensity. So for DNA, I think there's no problem looking at modifications. Yeah. You just have to have a good coverage. This is usually the problem. You need to have at least 24x, I would say, for methylation in DNA. Yeah, this is usually getting a good coverage along the whole area of interest in the genome.
Do you think that in the future it will be possible to look at RNA modifications and RNA structure using one sequencing data?
No, I don't think so. I would love to be wrong, but I think if you do a probing-based approach, you cannot distinguish what is from the probe and what is from the modification, and it makes it too messy. The other approach to look at structure is to look at the speed of the motor protein, and sometimes different structures are able to change the translocation speed. But the thing is, with new chemistries, they really are producing better proteins, the nanopore and the motor protein, that can account for structure and unwind very well and keep the translocation speed very even. So I think it's probably not possible, but I'm also not working in structure, so maybe I'm just pessimistic. I don't know. What do you think, Ivan?
Yeah, I agree. I think that most experiments that look at structure rely on probing the RNA, so it'd be hard to infer anything without probing previously.
Yeah.
There are a few questions from before the talk, so maybe I can just pick a few that were not covered by the other questions from the Q&A section. So what is the length of the RNA that can be sequenced?
So the upper limit, there's really no limit in theory. Of course, there is always an actual limit. For DNA, it's like 100, you can do over 100 kb. For RNA, I don't know what the longest that they've done for RNA is. But yeah, I do think it would be difficult to sequence a really long RNA, like Titan mRNA, I think is 100 kb. maybe you can get one read, but it's very, very difficult. Also, you have to have very good library preparation that's keeping things intact. So I would say that, yeah, you can sequence very long things, but there's a trade-off with coverage. I'm extrapolating from DNA, but with DNA sequencing, if you sequence something over 25, 30 kb, you tend to get some blockage of the pause, and then you have a reduced coverage. So you kind of have this trade-off. Like this ultra long sequencing kit from Nanopore, you can get 100 kb reads, but you have a coverage of 1. So there's that. But I think 10 to 20 kb you can probably do pretty comfortably. So maybe you could get exist. I don't know if someone ever did the whole exist mRNA, but if you were really good in the bench, probably you could. And then on the other limit, I think we published 75 or down to 50 kb, and I think we're down to 20 kb. And then also Nanopore are releasing or have released an update to the software where you can, so they realized after we published this paper that they are filtering out all of these short RNAs, and they changed their software, Minnow, so that they keep everything really above 20 kb. So that's kind of the range. Yeah.
Cool. So maybe 2 last questions, and then we can close today's session. So there is quite a few different modifications present in tRNAs. And what are the modification types that you can identify in tRNAs? And can you identify large modifications of tRNAs, such as granulated uridines?
Very nice question. So when I was doing this, I could do, oh, now it's been a year. So pseudouridine, M5C, M1A, depending on the context as well, what spaces are around. Sometimes you could, sometimes you couldn't. What else? Inosine, and a few others that I don't remember. And a lot of people have been working on this since I left the lab, and continuing the work with tRNA. And I think maybe they're up to like 10 different modifications that you can detect in tRNAs. And also there's a group in Japan, Tom Suzuki. He's also published a paper about being able to detect, I think, up to 10 modifications in tRNAs. So I think this will improve. I mean, it's pretty new that we're able to sequence tRNAs, so I think this will get better and better. In terms of large modifications, in theory, yes, we should be able to do that. Like I think we were looking, for example, at things like glycosylated versions of modifications, like on Q-seen. So I guess they're pretty big modifications, and we could do that. The thing that can be challenging is you need to, when you're developing these methods, have some prior knowledge. So you need to have a way to really confirm if what you're seeing is a change in that modification. So usually a knockout line or mass spectrometry. I think I read this question. It was also related to standards in mass spec. This can be challenging if you don't have a standard, but I think there are ways to also get around this. I know that when we were interested in the glycosylated versions of Q-seen, or whoever was interested in this, I think she worked with the mass spec facility to have a way to be able to check this in tRNAs, and then have a way to knock out or prevent Q-seen, and then confirm it with nanopore sequencing data. But I think it's always worth a try, and if not, maybe write to Eva and see if she might be willing to give it a go. She's usually pretty curious about such things, so I would encourage to email her.
I agree. That's a good idea.
And yeah, maybe Ivan will do it.
Or maybe I will do it. Yeah, feel free to email Eva. And as the last question for today, let's finish on a software-related note. So which software do you recommend for analyzing nanopore RNA data, as there are several different packages available in publications?
Yeah, so we and others published a lot of tools in the last couple of years, right? And this was because there was not really anything available. Nanopore hadn't published these tools, so we were doing what we could and whatever. So there are many tools out there, but I think that now Nanopore do a very good job of this. They are working on modification aware base callers for RNA based on the current intensity and not base calling errors, and they have this integrated for probably 5 modifications now, but the list will improve in their epitome workflow. And this makes life much easier if you can do it like that instead of using tools that may not be discontinued or not. I know Ivan and I have both had experiences with using Nanopore tools that were developed by a research lab that then were discontinued. We, I think, do a very good job of continuing tools, but it's not perfect, right? I mean, if you're a researcher, you publish something and then you also have to spend time maintaining a tool that you developed 5 years ago. You don't really have the time or resources for that. So I would go for Nanopore tools. I think they took a lot of what the community has published and realized what's needed and how to do it. And probably were also working on these things before that, right? But now they have tools that are very good that are going to be maintained and stand the test of time and will be tested on new chemistries and things like that. So you don't have to do all of this validation yourself. So this would be my recommendation. And now it's this Nanopore conference, so I'm sure they will announce new modifications. Yeah, so the list is growing, that's for sure. Yeah,
Perfect. Thanks a lot. I think here we can call it a day, and I will first like to thank you for the amazing presentation, and I will leave the floor to the organizers for the last farewell words.
Thank you so much. Thanks for an amazing talk and a really great discussion. Thanks for leading that, Ivan. Leading and participating. And thanks everyone for being here for that long. This was really great. As I mentioned earlier, this webinar is also being recorded and will be posted later in about 2 weeks, maybe. So if you want to revisit parts of the talk, you can do so. And thanks again, everyone, for coming. And I hope to see you again sometime soon. And thanks for, again, that amazing talk.
Oh, no, thank you so much. It was really fun. Thanks, everyone, for attending.
Bye.
Bye.
Bye.